As it was said, I'm Allison Kalinousky. I'm a human genetics PhD student at Johns Hopkins, and I will be discussing the ongoing clinical and translational research projects in Kabuki syndrome. So our end goal of research is to help improve the symptoms of patients with the disease through treatment. However, there are a few steps that we need to accomplish before we can move towards human trials. So just to kind of reiterate what Dr. Harris spoke about, first, we need to have a detailed understanding of the specific characteristics of the disorder we want to treat. Then we need to understand the mechanisms underlying the disease, because it's possible there could be overlaps with other similar Mendelian disorders of the epigenetic machinery, which could allow for common treatments among disorders. We also need to validate preclinical studies done in the lab to make sure we're seeing similar findings in humans that we're seeing in our model systems. And lastly, we need biomarkers and outcome measures that are specific to the disorder we are trying to treat, so that when we do conduct a clinical trial, we will have quantitative ways to measure improvement. So the first step towards moving to a full-scale clinical trial is to understand what needs to be improved in the lives of those with Kabuki syndrome. As clinicians and researchers, we have our ideas of what aspects of Kabuki syndrome should be treated, but we want to hear directly from those impacted by the disorder what is most important to them. There are a lot of different projects being done in the lab that have shown promising results, and my main interest is bridging the gap between lab work and the clinic. So to do this, I'm first collecting survey data on anxiety, behavior, sleep, adaptive function, and overall quality of life. These are areas that have been anecdotally seen by clinicians to be potential problem areas, which could potentially then benefit from therapeutic intervention. So this is a study that's currently ongoing, and it's open to both those with Kabuki syndrome type 1 and Kabuki syndrome type 2. It's completely virtual and takes about an hour to complete all of the surveys, and it's open to anyone that's over the age of 4. So we're also collecting data from siblings of those with Kabuki syndrome under the age of 18, which we're going to use as controls when we're analyzing the data. So all that would entail is just 2 additional surveys about the sibling. So if you're interested in participating, please send me an email. All you would have to do is fill out a consent form, and then I can send the surveys over to you via email, which can be completed at your home at your convenience. This study will continue till the end of November. So if you're interested, I encourage you to reach out to me as soon as possible. The next step of moving towards a clinical trial is to develop outcome measures so that we have quantitative ways to test improvement. Members of the lab have previously performed behavioral testing on a mouse model of Kabuki syndrome and showed that the mice have hippocampal memory defects. This was then tested in patients with Kabuki syndrome using tests designed to explore visuospatial function, which localizes to the hippocampus. The results from these tests confirmed what we saw in the mouse model, which helps to support the validity of this model. So as you can see from these graphs here, when comparing the results from participants with Kabuki syndrome, who are shown in red, to other people with the same IQ, shown in green, participants with Kabuki syndrome performed worse on the visuospatial categories, which is linked to the hippocampus. In other categories, such as sentence comprehension, participants with Kabuki syndrome are typically scoring above IQ match controls, but less than neurotypically developing participants, showing that visuospatial function is a specific weakness in Kabuki syndrome. So what we're currently doing is we're performing 2-day cognitive testing to build upon our understanding of these results. Since right now we kind of have a basic understanding of these deficits, our goal is to build a more comprehensive cognitive profile. This profile will help us create reliable and valid outcome measures that are specific to Kabuki syndrome, so we can test if we're seeing improvement after treatment in any of the areas that are lacking. This testing is done over 2 days to make sure that the scoring is staying consistent. This is important because if we use this testing for a clinical trial and we do see changes in the results after treatment, we know that it is due to the intervention and not to any variability in the testing. In addition to the cognitive testing, we're performing MRIs looking specifically at the hippocampus. This is because previous work done by the lab showed decreased growth of new neurons in the mouse model of Kabuki syndrome, in addition to that memory defect that I was just speaking about. This makes sense because the growth of new neurons is thought to play a role in memory and learning systems. So our goal of the MRI is to validate that we're on the right track with the other outcome measures we are creating through this cognitive testing, and also to develop biomarkers that are specific to Kabuki syndrome. Additionally, if we see a difference in the hippocampus of those with Kabuki syndrome, this will help support that the mouse model we're using is a good representation of human beings. So another potential biomarker is an ERP, which we're using to approximate a phenomenon called long-term potentiation, or LTP. LTP is when the connections between neurons become stronger with frequent activation. It's thought to be a way in which the brain changes in response to an experience, meaning it may be a mechanism underlying learning and memory. We think that people with Kabuki syndrome have differences in learning and LTP, which can make ERP a good biomarker for future clinical trials. It's been shown in mouse models of Rubinstein-Taybi, which is another Mendelian disorder of the epigenetic machinery, that these mice have deficits in LTP or this difference in learning. So as there are many similarities between Rubinstein-Taybi and Kabuki syndrome, we're testing whether this deficit is also seen in patients with Kabuki syndrome. So to do this, we're performing ERPs, which is basically a less invasive way to approximate LTP in humans because we can't do the same testing that we do in the mice. So for the ERP, it's very simple. We just place a cap with electrodes on the participant's head and then record their brain's response to sound stimulation. And in the future, we also plan to test the Kabuki mouse model to see if they also have these LTP deficits. So for these studies, we're currently accepting only those diagnosed with a mutation in KMT2D gene or Kabuki syndrome type 1 and who are over the age of 13. So to participate, you must travel to Kennedy Krieger, where you will complete 2 days of cognitive testing, an MRI, and an EEG. And in the future, we plan to expand to include patients with Kabuki syndrome type 2. The last step to move towards a clinical trial is to conduct a pilot clinical trial to make sure we're seeing effective results before moving large scale. Previous lab work has found that when the mouse model of Kabuki syndrome is put on a ketogenic diet, we see that there is postnatal rescue of the defects we see in memory, which basically means that the mice were performing better on the behavioral testing after being put on the diet for 2 weeks. We also saw that the mice had improvements in the growth of new neurons post-treatment. So we believe this diet is helping to restore the imbalance between open and closed chromatin that is thought to be present in Kabuki syndrome. Our pilot clinical trial puts participants on a modified Atkins diet, which is basically a slightly less strict version of the full ketogenic diet, as it allows for more carbs and it does not monitor protein intake, but we do know it's just as effective as a ketogenic diet. And we're testing to see if we see similar improvements in people as we did in this mouse model. Another thing that we know is that those with Kabuki syndrome have a distinct methylation profile, which is a pattern of methyl groups on DNA that controls which genes are expressed. This profile can be used to diagnose Kabuki syndrome instead of having to sequence someone's genes and look at their changes. So during the pilot trial, we are collecting blood at the 0, 6, and 12-week mark, which we'll use to run a methylation array, which is basically just an experiment that we could look at this methylation profile. From this, we want to see if this signature changes after treatment, which could indicate that the imbalance of open and closed chromatin that is seen in Kabuki syndrome could be altered after birth. If we see some of this balance restored while on the diet, it could potentially correlate with symptom improvement in Kabuki syndrome. Additionally, during the diet trial performed on the mouse model of Kabuki syndrome, the lab collected urine from the mice at the 2-week mark and found that the urine contained elevated levels of beta-hydroxybutyrate, or BHB, which is a ketone body that is produced during ketosis. And this level was higher in comparison to control mice who were also placed on this ketogenic diet. They then placed a mouse model of Rubinstein TB on the diet and found that those mice did not have this elevated urine BHB level and instead had levels more similar to the control mice. So this elevated level is therefore unique to Kabuki syndrome, indicating there might be a potential metabolic defect associated with Kabuki syndrome. So based on this mouse model and combined with other published data, we're beginning to investigate whether there are metabolic abnormalities in Kabuki syndrome. So during our pilot study, we're collecting urine from participants at the beginning and end of the diet in order to see if participants also display this elevated level of ketones. So to participate in this 12-week diet trial, you must be over the age of 18 and diagnosed with Kabuki syndrome type 1. It requires a visit to Krieger at the beginning and end of the diet, where you will perform cognitive testing as well as have a blood draw and urine collection. You also will get a blood draw at weeks 3, 6, and 9, but this can be done locally and you do not have to come back to Hopkins for that. And then during your first visit, we provide you with all the information needed on how to follow the diet. And we even have an app to track your carbs and to provide you with ketogenic friendly recipes. So just kind of to summarize everything that I've spoke about, there are currently interesting preclinical leads, but we first need to figure out what's important to patients with Kabuki syndrome. We're also actively seeking to better understand Kabuki syndrome and to develop outcome measures for future clinical trials. And lastly, we're conducting a pilot clinical trial based off of promising data seen in the mouse models. So we're so grateful for all of those who have already participated in our research. It has the potential to directly lead to therapies in the near future. So it's important to get as many participants as possible. So if you're interested in any or all of the studies, please reach out to any one of us. This is our study team here. I've listed all of our contact information as well as a summary of the eligibilities for each study. We also have flyers about our studies that are posted in the research recruitment tab for you to access after this conference. I hope to be hearing from a lot of you soon, and thank you for your time and the opportunity to speak today.